Hello, everyone, and welcome to our first session. I am Nate Ritz, a microbiota gut-brain axis researcher and postdoc at the Venkatesh Lab here at the Institute for Systems Biology. In this session on neural effects of gut microbial metabolites, we will explore how the gut microbiota impacts distant organs, like the brain. And for this, I'm excited to have two excellent speakers here, and Dr. Brittany Needham and Dr. Giada De Palma, to present their very exciting work. First up, we'll have Brittany, who is currently an assistant professor at Indiana University School of Medicine and Stark Neurosciences Research Institute. She studied mechanisms of bacterial pathogenesis that can be utilized as tools in vaccine development at University of Texas at Austin, where she earned her PhD in the laboratory of Stephen Trent with a new focus of the gut microbiota, and in particular, how gut microbes can influence the function of the brain. Brittany completed her postdoctoral work at California Institute of Technology with Sarkis Mazmanian, and now her lab focuses on the dynamic metabolic activity and bidirectional flux of molecules between gut microbes and the host, where her group is working to elucidate specific mechanisms mediating gut-brain interactions that remain poorly defined. Her lab aims to improve understanding of the molecular communication between gut microbes and host systems to uncover critical features of gastrointestinal, immunological, and neurological diseases. And recently, she was recognized as a finalist for the 2024 Nostern Science Prize for young investigators researching the microbiome. So now, without further ado, please join me in virtually welcoming Dr. Needham for her talk, which is entitled Brain-Relevant Microbial Metabolites.
It's a pleasure to be here. Thanks for the invitation, and it's nice to talk to you about this work today. I will focus on our work studying kind of gut-brain interactions and talk about some of the published work we've done as a kind of a background and context, and then where that work has led us and what we're currently doing. Okay, and I think the audience is probably mostly made up of people who are very familiar with the gut microbiota, so my introduction will be brief and really just to contextualize what I'm talking about. And really this electron micrograph image is something that inspires a lot of thought in my lab. It's a very simple image, but just to remember how close bacteria and other microbes get to our epithelial cells. You can see there are bacterial cells and these epithelial cells along the edge, and they're really close, and it's easy to imagine all of the bidirectional flux of chemicals and byproducts and host and microbial influence on each other. And so we think a lot about that in my lab, and we think about what can we do or what can the microbes do to influence these phenotypes and this interaction. So of course, a lot of the things that we do, or a lot of things about us, change and influence the microbiota, like diet, age, our lifestyle, medications we're taking, as well as the other reverse, the microbes affect us too. Without our microbiota, of course, we would not have a normally developed immune system. We would have a harder time with nutrition. Our microbes really help with a lot of breakdown of complex plant polysaccharides and other food products that we get a lot out of due to our microbes. And of course, they protect us from invading pathogens. And something we think a lot about is how that's important for the proper development of our nervous system. All of those things combined and these functions that our microbes play on our bodies and on our kind of the whole host. Okay, and there's a lot there. So I think we could probably have 100 speakers in this webinar and all of us looking at some different aspect of those complex interactions. We have a very complex host environment, and then we have this jungle of microbes living inside us that are also bringing in a lot of complexity. So in my lab, we really think about the small molecule metabolites, or these byproducts that microbes make, and those of them that get into our bodies. We think of that in a lot of different levels. So at the substrate level, so these microbes with massive genetic diversity and many enzymes that are evolutionary divergent from our own enzymes are in contact with many different molecules. What we eat, molecules that our bodies produce, molecules that other microbes that are in the gut are producing. So on the left, you can see there's just a lot of categories of molecules that the microbes are coming into contact with. And then we have so many enzymes in our gut microbiota and the microbiome that encode basically a huge capacity to modify these molecules into an even more diverse pool of molecules that can get into our bodies and exert many of the mechanistic bioactive results that we think about when we think of gut host or gut brain interactions. So I see that as kind of an exciting challenge to kind of figure out which of these molecules matter in which way to our bodies. Some of them are harmful, some of them are beneficial, and really it gives us 3 areas to look at for therapeutics. We can change what we put into the system that microbes come into contact, change the enzymes that are different from ours. We might have fewer side effects if we alter those enzymes, and then we can also try to change the output. What are these microbes making? How can we alter that for our own host health? And as Nate introduced me, and as I mentioned, we're doing this in the context of looking at the brain. And there are a lot of neurological, neuropsychiatric, neurodevelopmental conditions that are associated with an altered gut microbiota. And in turn, there have been an increasing number of studies showing that manipulating the gut microbes in some way, whether that's gnotobiotic animals or antibiotics or probiotics or other treatments, altering those microbes then also play causative roles on some of the features of these conditions. And I've listed here some of the ones that are the most well studied in these contexts and showing the most causative phenotypes thus far. And we had one where we just really wanted to ask, can we identify something specific in these interactions between one of these neurological conditions and the brain? Like the gut and the brain, how are these things happening beyond a correlational study and beyond something that's just associating a change in the gut and a change in the brain? So that's really what we aimed to do when we set out on this study. And I had access to some CHARGE studies, it's called, some samples from the CHARGE study. And these are samples from neurotypical controls and these are human patients, samples with altered neurodevelopment. So we have serum and we have matched fecal samples. And we did what many of us do. We did some omics and we looked at every small molecule we could measure in these plasma, I think I might have said serum a second ago, plasma and fecal samples. And this was as untargeted and global as we could do, but I was interested in looking at what microbial molecules we could pull out. And the molecule that was the highest fold change in all out of everything was a molecule that is only made by the gut microbiota. Our mammals don't have enzymes encoded to produce this molecule, and it is called 4-ethylphenyl sulfate. So it was we saw a high fold increase in these samples with altered neurodevelopment. And when we did a machine learning random forest analysis, it was also one of the top molecules that showed up as differential between these two groups of individuals. So that gave us a lot of maybe reason to keep studying this molecule. We also had some preliminary work from Elaine Shao and others in the lab that had shown that this molecule might be interesting. So really it's like kind of coming from 2 prongs, maybe it's worth studying this molecule. So we engineered bacteria to be able to produce this molecule. So this is an engineered pathway that is made from microbes, but it is not necessarily a pathway we've ever observed in any single microbe. This was just for a proof of principle and to be able to really study it in a targeted way. And you can take tyrosine and microbes will break that down into 4-ethylphenol, which is here, and that's as far as the bacteria take it. And then when it enters the host system, it quickly gets sulfated very efficiently into 4-ethylphenyl sulfate, or 4-EPS. So you'll see on my slides 4-EP, 4-EPS used slightly interchangeably, and that's because the bacteria are making 4-EP, but the host is mostly exposed to the 4-EPS form due to the efficiency of that sulfation. Okay. So once we engineered that pathway into some bacteria, we had a tool to control study that in mice. We take germ-free mice and we put either 4EP minus, so microbes that can't make 4EP, or another group of mice that are just wild type. Well, these are all wild type mice. We colonize them with 4EP producing bacteria. And the bacteria are isogenic in every way besides the difference in this pathway. And we colonize them. It's stable colonized for as long as we've tested. And so it just sticks around and is either producing the molecule or not. And then we can confirm that these mice are then exposed to 4EP in the feces or 4EPS in circulation. I'm showing urine here. It also gets into the brain and in the serum. And we don't see any of it in mice that shouldn't have it, in our controls that are colonized with the control bacteria, and in the presence of the bacteria producing that, expressing this pathway, we see high levels of 4EP and 4EPS being produced. And we looked at these animals in a lot of different ways, but because I was interested in the brain, one of the first things we also looked at was just to ask, is there any difference to the brain in general, just due to exposure of this, exposure of the mice to this molecule through their gut bacteria. So we did something called functional ultrasound imaging, which these are mice that are anesthetized, but they are alive and their brain is functioning, and we can measure their cerebral blood flow through an ultrasound. It's really just a probe. It's extracranial. It's an ultrasound just like you might be familiar with from your own life. And we can measure blood flow in various regions of the brain. And then I can see on the left here, I've overlaid a brain atlas, and we can arbitrarily assign a number to each brain region and then make these correlational matrices between patterns of brain activity in these different regions. So what we're asking here is there any correlation between the amount of blood flow and the patterning of blood flow that we're observing in one brain region versus another as a metric of functional connectivity? So we do that. I won't go into more detail here, but just you can kind of see by eye that this bottom row here, the mice exposed to 4EP have higher levels of functional connectivity. You see more of this warm coloring when they were exposed to the molecule. And then we're able to go back and look and, what brain regions are the pairs that are explaining this data, and which brain regions might be interesting to follow up on. So we went a little bit further and did a more high resolution study. This is autoradiography, where it's our same two groups of mice that are 4EP minus and 4EP plus, but we've injected a radioactive tracer. It's a 2-deoxyglucose, and it can't get broken down. So when it gets taken up into the brain, it gets trapped there and not metabolized, and cells that are highly active are going to take more up, but more will get trapped. So we do that, we can measure the difference in brain activity between our two groups. So what I'm showing on the right is between the two groups. This is a summary data of many mice, and the warm colors indicate a higher brain activity in regions where more of this uptake was taken. Okay, so we see, and again, I won't go into a lot of detail about the brain regions here, but we do see some brain regions that we were excited about: the amygdala, the BNST, the regions of the hypothalamus thalamus, and these are regions that are known to be involved in emotionally related behavior, some fear responses, and anxiety-like behavior. And the reason that was exciting, because at the same time we were also doing behavior testing and observed, we just really ran them through the battery of all the behavior tests, and anxiety-like behavior seemed to be the behavior that most consistently and strongly showed a difference. So mice that were exposed to this molecule, 4EPS, have a decreased tendency to explore an exposed region. I'm showing that here in elevated plus maze on the left and an open field in the center. The mice are less likely to explore the end exposed regions of a raised maze, or to enter the center exposed open well-lit region of a box that they are in, that we're videoing them. And they're also more prone to do kind of anxiety-associated digging when they are put into a cage with this grid of marbles we're showing here on the right, marble bearing. Marble bearing can also be used as a test for some other behaviors, but because we don't see, so for example, repetitive behavior, but we don't see phenotypes in other repetitive behavior tests, so we think this marble bearing really isn't associated with their anxiety-like behavior. So we see as a result of this molecule being produced by gut bacteria that we're seeing changes to brain patterns and changes to complex animal behavior, which is really exciting. But as I told you before, our goal was not only to pinpoint that a phenotype is specifically resulting from a single signal from the gut or from a specific signal, but also to kind of try to work out the mechanism of how. And I won't have a full answer mechanism for you today because we're still working on it. But we did a lot of experiments trying to figure out, okay, what's between? We have the gut, we have the brain, we have these phenotypes. We know that they're causally related, but how is it happening? And so through some RNA-seq and some other experiments, we were led to the conclusion that oligodendrocytes are the brain cell that is influenced by 4-EPS and causing this phenotype and the behavior that we're observing. So of course, oligodendrocytes are the cells of the brain that produce myelin. So as they differentiate from precursor and early oligodendrocytes into mature myelinating oligodendrocytes, they begin to wrap their membrane around neuronal axons and provide insulation for neuronal firing. And they're really important. There's a lot of interaction. It's a very dynamic and intimate relationship between oligodendrocytes and neurons, and a lot is known about neuronal firing and a lot is known about oligodendrocytes, but the field now is starting to learn even more about how much that interaction is important beyond just structural kind of developmental establishment of myelin, but that myelin and neurons have a lot of crosstalk and interaction throughout life. In fact, even throughout adulthood, we continue to have plasticity and modification to our myelin. As we learn new things, have new experiences, we have this remodeling of myelin that continues to occur. So it's an exciting kind of target for us to be looking at that the gut might modify, because these are really cool cells, and I didn't know a lot about them before we got these results, and it's a really exciting cell to study. So here's another image of one. So you can see this is one of our ex vivo slices that we cultured, and you can see red. The red's almost covered up, but there's an axon here stained with neurofilament, and then you can see the green kind of laid on top. And that's MBP, a myelin protein that we're staining for. So there are a lot of ways to study these in vitro, and we weren't the first one. So when we first got these results, we observed that there's a previous paper from John Cryan's lab where they were looking at germ-free versus control mice, and they observed that germ-free mice in some brain regions, especially in the prefrontal cortex, had this really thick layer of myelin. And so there was indication that, okay, maybe something in the gut could be affecting the brain. The myelin in the brain. And also antibiotic treated mice, when it's just like a cocktail of antibiotics and you've really depleted the microbiota, there are differences to oligodendrocyte cell differentiation. So it's not completely crazy that maybe 4-EPS could affect myelination in oligodendrocytes. So we went further to characterize this, and I'll just show you this one image here. This is the corpus callosum. So this is a region of the brain where axons are in parallel. And if you imagine them coming out of the plane towards you and you take a cross section, these dark gray rings that you see are myelin layers that are around axons. You can visually see a striking difference between 4-EP minus and 4-EP plus. And we did a lot of these, and we looked at a lot of images, and this was a phenotype that consistently showed up across animals and across images, that we have not fewer axons in the 4-EP plus mice, but fewer myelinated axons in this region of the brain. And the myelin's also a little bit thinner, and there's some other phenotypes that are much more mild compared to demyelinating disease like multiple sclerosis, but some obvious and consistent changes in myelin. So we are still now working on figuring that out. We've done a bunch of MRI. We're really trying to figure out what regions the myelin is the most different, and precisely what is 4-EPS binding in the brain. We know it gets in directly. We know it gets in, and we know that it can directly cause this phenotype, but we're trying to work out what is the protein target. We have some hits that we're working out. So another end is So exactly how's it happening in the brain? And the pathway that we're using is pretty artificial. So we're also interested in what is the actual natural context of 4AP and 4APS production. Any gene that we used for our engineered pathway is present in about 25 genomes, like in common human gut microbes. And so we know that the genes are prevalent, but we haven't identified a single microbe that has all of them. And when we modify the diet and change tyrosine levels or change other dietary aspects that microbes could use in a mouse with a full microbiota, we sometimes get very surprising results of how much 4APS can be made. We tried to eliminate all the tyrosine, which we thought was probably the primary substrate to make 4APS, and in that diet, when we had removed almost all the tyrosine, we actually got an increase because the thing that we had replaced it with was soy protein, and it has other things that can be metabolized into 4APS. So that's kind of something we're working on currently. And the reason you're probably wondering why I have a person with his nose in a wine glass at the top of my slide. That's just to, as a side note, that 4AP is really smelly, and it's something that can be a contaminant of wine, and that's relatively well characterized. And actually, this reference here, the Australian Wine Research Institute did a study asking, like, how good are people at detecting this contaminant in wine? And we are very good at it. We can taste it when it is in wine and it's contaminating. It's gross. And when we work in the lab with it, we usually give people heads up, like, I'm getting some 4AP out. It's going to smell like, fair warning that it's not pleasant. Okay. And then we're also trying to figure out what microbes might degrade it. Why is it being, not why, but what are the results of 4APS production in the gut? How does it affect other microbes? Are there microbes that degrade it, and could those be beneficial to use therapeutically to reduce these levels of 4APS? Okay. And then one final part of that story, before I show some of what that has led to, is that we were able to do a preliminary look in preclinical model, the same preclinical model I've described thus far, and also a phase 1 clinical trial exploring whether we can reduce levels of this metabolite directly from the gut and keep it from being able to enter circulation and then limit how much it gets into the brain and whether that will have an effect. So we did that with an oral adsorbent. It's called AST-120 in Japan. It's approved for use to limit molecules like this from circulation in chronic kidney disease patients. And in our study, it was named AB 2004. And when we administer this drug to mice, we did see decreases in 4APS levels in our mice, and we did see improvements in the phenotypes that I've described thus far in our mice. And so we did this phase 1 clinical trial. It was a small trial. Everyone got the drug, and everyone knew they were getting the drug. So it was open label. And for 8 weeks, they got increasing amounts of this drug that we hoped would reduce levels of 4APS and other molecules that are structurally very similar, and hopefully reduce some of the anxiety-like symptoms of these individuals. These are teenage boys. Okay, so if you see on the left here, orange is baseline on the left, and then these are 4APS levels. And at the end of treatment, almost everyone had much lower levels of 4APS as a result of this oral absorbent. And you eat it and it just passes straight through. There's no absorption of the body of this drug. They're small little beads that really just pass right through. And we also saw decreased levels of anxiety scores and irritability scores in these individuals. And again, I can't state enough that this is preliminary and kind of a first look, but and I should also say that I was part of this study, but I did not run this full clinical trial. This was Axial Therapeutics, and did all of the hard work with the clinical trial and recruiting people. And I really just got to benefit from helping design the study and then get to look at all this cool science that came out of it. Okay, so what we're kind of looking at now, beyond trying to figure out the mechanism of 4APS, is the idea that it's unlikely that 4-EPS is the only metabolite from the gut that can affect the brain and affect myelination in this way. And so we look at these images, like here's a control mouse. This is, I'm just showing you one axon, so take that with a grain of salt. But here's one axon and this level of myelin in a control mouse that is a standard wild-type lab mouse with a conventional microbiota compared to a germ-free mouse, which is much thicker myelin, excuse me. This is a more drastic difference than what we're seeing just with 4-EPS. On the right here, so this is a longitudinal axon, and with 4-EP, we can even like count the rings of myelin. It's thinner myelin on the 4-EP compared to 4-EP minus, but it's not as drastic as this difference as you would expect, the difference between a full microbiota and totally germ-free mice. So we're really interested in what other molecules might be involved, what else might be playing this role, and if we can identify more of them, maybe we can use them in combination to really modify this, modify myelin patterns in the brain. We can remove harmful ones in the context of disease. Maybe we can find some that are beneficial to promote myelination, to be helpful in demyelinating disorders. And so thinking back to this pool of metabolites and kind of how do we do this? And one way that we have done it is we did a screen where we took a lot of the gut-derived metabolites that are commercially available or not too difficult to get synthesized, and we incubated them in vitro with oligodendrocytes and neurons and tried to look for some that had an indication that they might be involved in affecting either oligodendrocyte differentiation or myelination. And I'll just show you some very preliminary data that is not ready for prime time yet, but we have one of these metabolites that came out of our screen. We've gone further with it to then take it in vivo. We've engineered bacteria that can produce it or lack it and put it into germ-free mice, just like we did with the other metabolite. And when we do that, we can see that, just for simplicity, I'm keeping all of this very general. In the cortex, we have decreased levels of MOG and MBP, which are both markers. These are proteins that are involved in part of myelin. We see decreases of that. And interestingly, again, these images, these are early days images, and this is consistent across many images of this pilot study we've done, even though I'm realizing these are kind of blurry, but in our control mice, we see thick myelin. Like the layers of myelin are all compacted appropriately and right next to each other. And in our mice where we've exposed it to this metabolite, and I'm not sure if you can see it here, but we see some decompaction consistently across that. There's a lot more space between these lamina of myelin. And so that's really interesting early phenotype that we're following up on, and we're doing a lot more work. But that was enough to embolden us to say, like, okay, well, let's look at these mice and some MRI. And so we did myelin water imaging, which is similar to DTI, if you're familiar with that. We're looking at the trapped water that is within myelin. And if you're looking at this webinar on your phone, then you're really only going to be able to see this first image. But I've just tried to show a summary like this. There are all the sections through the brain, and you can see anywhere that it is blue, or in this top image, anywhere that it is colored at all. We didn't see a single instance, a single voxel of decreased trapped water, so decreased myelin in our metabolite exposed mice, which was interesting because that is what we expected. We thought we were seeing a damage of myelin in our other studies, and that we would see a much decreased myelin level by MRI. We see the opposite. So anywhere that there's a difference, we're seeing increased trapped water, which can be an indication of increased myelination. But with our EM images, we're now trying to figure out, is it just that there is altered myelin, maybe more trapped water, some decompaction happening? And these are early days and just an exciting thing to be working on still, and trying to figure out now what's the mechanism of this other metabolite. And in the future, we can combine these metabolites and look at them together and see, like, what are the effects of these pools of metabolites? Because in real life, we will never be exposed to just a single metabolite in our bodies. And we don't necessarily want to remove them completely either. So until we can figure out these mechanisms, we don't know when is it beneficial, when is it harmful, and kind of how should we modulate it in a clinical stage. So that's really kind of big picture where we're at with trying to work to be more mechanistic between the gut microbiota, these gut signals, and how it's affecting the brain. And ours has been very focused in on myelination and these oligodendrocytes, and we're working to figure out where in the pathway of differentiation of oligodendrocytes as they become mature myelinating cells from these early precursors, where in that pathway are these metabolites hitting it and causing these changes. And this work has been done by, some of this was as a postdoc, so I've listed my postdoc advisor here under collaborators, but he's instrumental in all of the early part of the work that I described, and my current lab members that are following up on this work and also expanding it to these other metabolites, especially Jamie Nolan, who is doing the second half of what I talked about. And also like to thank our funding sources and all of you for listening, and I am happy to take any questions.
All right, excellent. Thank you, Brittany. That was a fascinating talk. Yeah, we have time for a couple questions, and just to remind everyone, you can post your questions in the Q&A. And then you could actually also vote on what questions everyone likes the best, and that'll kind of bring it up in the rankings. Additionally, we'll have a panel discussion at the end of the symposium, and especially if it's questions that regard multiple different types, you can bring them up then as well. But yeah, first up, let's see. How does creatinine interact with 4-EPS? I see that a graph for urine concentration of 4-EPS is corrected for creatinine.
Oh yeah. Um, we we want to control that some mice haven't been drinking a lot of water and some mice are more dehydrated. And so creatinine is just a measure of hydration, really. And so we're just using that to normalize that we aren't artificially observing an increase in 4-EPS levels because those mice drank a whole bunch or drank very little. And so it's just really a normalization tool.
Cool, great. Next up, have there been any integrated multi-omics data analyses done to ascribe what are all the specific microbial communities that participate in the production of 4-EP?
I don't know that anybody's done that well yet. We've played around with it a little bit, and it's a bit of there's we know that there are several pathways that microbes use to make the molecule, and so I think that we know enough now to really start a really nice study doing that. It's not really my expertise, but I think it's a good time. The field is, I think, has gained enough information and tools and technical expertise to be able to do that, but it hasn't really been done.
Gotcha, gotcha. So to be continued there, kind of.
Yeah. We've mostly looked at kind of pinpoint level of like, let's look for these enzymes or this pathway. We haven't done a lot of very broad omics level look at that.
Okay, cool. Next up, thank you for the great presentation. I was wondering whether 4-EPS is thought to be a cause of the neural defects or a consequence of them, kind of a chicken and egg question. And then there was a part two to that question, which is what is the most impactful way to reduce 4-EPS levels? In your talk, you bring up the adsorbent AB, was it 2004?
Mhm.
Um, have you also looked at using like a different, either a probiotic or some sort of microbiota targeted therapy to kind of approach that question?
Yeah, we haven't done that yet, mostly because almost everything I presented was very reductionist and lab work. Like we really needed to pinpoint that it was specifically this molecule. And so there's a lot of work now to go more broad and say like, okay, well, in the real world, when is this molecule good versus bad? I can say that we've measured samples from probably over 500 people now in different contexts and studies and ages and comorbidities and other kind of metadata about these individuals. And we've never found a sample where somebody didn't have any levels of this molecule. So I think that it's probably a very normal product of our microbiota. But that perhaps there are instances, either as a result of a disease or as a result of diet that there's a higher level. And whether chicken and the egg questions are always difficult. I don't know. We know that it exacerbates or can cause these problems, but whether the higher level of 4EPS is a result of an underlying neurological change or a change of a behavioral change, like if you have an altered diet, or, like why does the level at first go up? I don't know, but we do know that this higher level, there's a population of individuals that we have that seem to be above this threshold where it might be a damaging molecule. But again, in real world scenarios, we know very little about how bad or good this molecule is. Maybe it has some roles that are beneficial.
Right. It gets much more complex quickly in the
Yeah.
in the hospital setting, right? Or real world. Okay, well, great. I think we'll have to leave it there for this talk and now move on to the next. But again, thank you very much, Brittany. That was an excellent talk.